Hello everyone, welcome back to another session of RNA biology. So we were here in the previous class that X chromosome inactivation and what is its take on RNAi. So we know T6 and X-Cyst, they follow exactly the principle of RNA interference, and let us dissect it out, and these details are taken from this paper published in Science, that is intersection of RNA interference and X-inactivation pathways. So here this figure you can see it, the pre-X chromosome inactivation, XCI means pre-X chromosome inactivation stage. You have a situation that X-Cyst expressed, T6 expressed, and if both are expressed together, we can understand that both will be degraded. That is before the X chromosome inactivation. That means both chromosomes are active because there is no weightage or no bias or no timing bias given to one of them. Now let us classify this into two. One is XA and another is XI. You will know what is XA. XA means X active, active X chromosome. XI means X inactive. So X active, we know that T6 has to be expressed and X-Cyst is not preferred. So what it should do is it has to create some local effects that is mediated via transcriptional gene silencing, and this has to make sure the transcriptional gene silencing has to make sure that X-Cyst is not expressing in the XA, means X active. If TGS is not working on X-Cyst, then it cannot remain active. It will go automatically inactive. So the local effects of X-Cyst T6 interplay make sure that X-Cyst is not expressed in the active X chromosome. On the other hand, X inactive, what happens? X-Cyst induction happens and PRC2 recruitment of H3K27 trimethylation causes a global silencing, and this creates the compaction of the entire chromosome, that is XI, X inactive, and this will lead to the inhibition of all the entire chromosome. So gene expression will be prevented, only the coated X-Cyst stays there. So X-Cyst induction ensures the global change, unlike what you see in the case of T6 expression. T6 do not bring in any global change. It bothers only the transcriptional gene silencing of X-Cyst. It bothers only the blocking of X-Cyst. It do not bother about the rest of the chromosome. Whereas if X-Cyst is expressed, it has to make sure that the entire chromosome is off and the coated X-Cyst stays there. So for entire chromosome inactivation, it depends PRC2 recruitment and H3K27 trimethylation and triggers a global silencing, and rest of them you already know, a specific histone isoform, macron H2A recruitment, etc. has to happen. Now let us see X-Cyst transcription in ES cells, embryonic stem cells. So in undifferentiated cell, undifferentiated means it is stem cell remaining as stem cell. You can see both two dots that is green in color. So how this is done? This technique is called fluorescence in situ hybridization, where you take the probe. What is the probe? X-Cyst RNA is the probe. You label it with a fluorophore and you hybridize it in a denatured condition onto the chromosomes or onto the nucleus. So this entire circle, blue color circle, is the nucleus, and in the nucleus you can see two dots, and these two dots indicates there is no weightage of X-Cyst over the other. It is equal, but it is low, and you can say low because looking at the neighboring pictures. Neighboring pictures, once it is differentiating early stage, you can see huge expression of X-Cyst in one of them, and you can guess it, that is going to be inactive, whereas moderate or nil expression in the other chromosome, that dot remains more or less the same. And as the differentiation continues, you will see this is disappearing completely because you don't have even tiniest expression of X-Cyst, and the high level of expression continues, because even the tiniest level of X-Cyst expressed, that will cause the inactivation. Even if it is not complete, it can cause partial inactivation, so you don't want that to happen. So eventually the one which is having high level of exist will remain inactive. The one with low or nil expression, which is supposed to be somewhere here, it is missing. Exist expression is missing means it is an active chromosome. So this you can prove it by fluorescence in situ hybridization. It's a very powerful technique used in RNA biology. So maintenance of the silent X chromosome. So like I told you in the earlier class also, making a 10-storied building is easy. Easy means possible, but maintaining that is really tough, because it can go dirty. If it is not maintained, it can go really bad. Same way, once X chromosome is inactivated, it has to be maintained. So many things has to be taken into consideration, such as replication timings. That is, replication of this inactive X chromosome happens very late in the S phase of the cell cycle. And nuclear compartmentalization. Normally it is compartmentalized in a place where the gene expression events are very low, normally towards the periphery of the nucleus. Gene expression events are very low. The inactive X is found usually at the periphery of the nuclei, where in general the gene expression events are very low. I am not saying that in the periphery there is no gene expression, but in a global way, if you want to make as a statement, towards the periphery gene expression events are low. It is almost like a discarded territory. So there you will have this X chromosome, inactive X chromosome located or distributed. So let us have a recap of what we have already seen. X chromosome inactivation. In mammals, the females carry 2 X chromosomes, whereas the males carry only a single X, giving a double dosage of X-linked genes in XX females relative to XY males. So this is the principle behind it. To balance the X-linked gene expression levels in males and females, female cells invoke a mechanism, what is called X chromosome inactivation or XCI, to randomly silence one of the maternal X chromosome during early embryogenesis. So it depends, later in the organism either paternal or maternal will be silenced. Prior to cell differentiation, both X chromosomes are made active. Both X chromosomes are made active. Now it is a question of randomness. During cell differentiation, each cell counts its number of X chromosomes. How many X chromosomes are there in a cell? If there are 3, 2 of them has to be made inactive. Only 1 is allowed to be active. One X chromosome is randomly selected for inactivation. The LNC RNA, long non-coding RNA exists, is upregulated on the future inactive X chromosome, termed as XI. A gradual chromosome-wide silencing process is then initiated on the XI, so that the entire chromosome is now compact. Once established, this silent state is possibly or it is stably transmitted through each round of cell division in a heritable manner. That means this will continue to be accepted and maintained. So X chromosome inactivation, looking further, during development, females undergo 2 forms of X chromosome inactivation. One is imprinted and the another is random. So imprinted X chromosome inactivation occurs during early embryogenesis. The paternal X chromosome, referred as XP, is preferentially silenced and it is maintained in extra-embryonic tissues throughout the development. Random XCI happens later. All imprinted epigenetic marks on XP are erased. That means paternal X are erased in cells of epiblast lineage, and these form future embryo epiblast cells. A second round of X chromosome inactivation is then initiated, where either XP or the maternal X chromosome, that is referred to as XM, is randomly silenced. No bias is given to any of them. Randomly silenced, and this randomness in chromosome selection is directly observable in the coloration pattern of female calico cats, which we have seen it already. Examples that we cannot say that black color is more or yellow color is more, or black color is in a particular shape or yellow color is in a particular shape. So it is absolute randomness. No design pattern will be followed. So it can be random patches, you can see. X chromosome inactivation is initiated from X-Inactivation center, which encodes 5 non-coding RNA of non-functions, and these non-coding RNA are X-Cyst, T6, Excite, Rep-A and JPX. So these are all the non-coding RNAs, and X-Cyst is upregulated only after transient pairing of 2 X chromosome. Transient pairing basically meant for counting, and pairing is the way in which the cell counts the number of X chromosome. So this is what we should know. Unless this pairing happens, you simply cannot count it. So depletion of any of these factors involved in pairing, such as CTCF and OCT4, they are important in the counting purpose, blocks the pairing of the cells with an aberrant number of active X chromosome. That means both the X chromosome can remain active or both can remain inactive. Usually it shouldn't happen if there is a imbalance happens in the expression level of CTCF or OCT4. These are all genes responsible for the counting. X-Cyst expression is regulated by T6 and in a negative manner, and Excite also affected in a negative manner. JPX affects in a positive manner, and Rep-A affected in a positive manner. So X-Cyst expression is stringently regulated by various supporting non-coding RNAs. So perinucleolar targeting of the inactive X happens during the late S phase, and it is maintained in the S phase, and it is always found in the periphery, and its replication happens in the late S phase. So evidence for a role in the maintenance of silencing is discussed in this article. So you can read, and we will also address it in a clear manner in the subsequent topics. So this is a very interesting article, so which is published in Cell a few years ago. So X chromosome movement in differentiating mouse embryonic stem cell. So we know that number of X chromosome during early phase, before differentiation happens, early phase of embryonic development, the counting has to happen. Once it is for counting, it has to pair. For pairing, it has to be close by. Once it is close by, then it slowly moves apart. And so pairing, counting, and the choice is decided, and 2 dots are close by, you can see here. And X-Inactivation center is what is marked in this green color dot, and then they slowly start moving apart, and later one of them will start expressing too much of X-Cyst we saw in the previous picture. So what happens? The X-Cyst RNA coated on X chromosome localizes near the nucleolus. So nucleolus is a place where you have too much of expression of ribosomal RNA. So this is stained, nucleolus is stained with a marker anti-fibrillarin. Staining is done for a protein, and that can mark, because nucleolus have got too much of expression of anti-fibrillarin, and that is seen in red in color. And you can see the X-Cyst RNA is very much close. So what it indicates, this X chromosome, X-Cyst coated X chromosome means inactive X chromosome, it comes closer to the nucleolus, which in turn eventually become closer to the periphery of the, say like here it is closer to the nucleolus, then it eventually located to the periphery of the nucleus. Nucleus, this is like a circular shape, like a oval shape, circular, and it is moving towards the periphery. And whether you do RNA fish or DNA fish, like fish stands for fluorescent in situ hybridization. You can use RNA as a probe or you can use DNA as a probe. In both the cases, you can see that its location is towards the periphery. So X-Inactivation center always have got a nucleolar association if it is inactive X chromosome, and if it is an active X chromosome, it is not having a nucleolar association. So this is something important to notice. So autosomes carry carrying the X-inactivation center are target to perinucleolar site. So if you are taking this X-inactivation center, that portion you are taking it from the X chromosome and you are inserting it onto another chromosome, which is autosome. Autosome means chromosome number 1 to, in case of human, chromosome number 1 to chromosome number 22 is called autosomes. So you take this X-inactivation center and put onto any other autosomes. You see that this chromosome now go closer to perinucleolar site. So this indicates X-inactivation center have something to do with association with the nucleolus. So you can see here this different location. This is inactivation center, exists T6, and you have XI, TSX, and many more genes are located. So this center, this region is what you are detaching and implanting onto a autosome. So autosomes carrying X-inactivation center are targeted to perinucleolar site. You can see this in this example. So bottom panel is the control. That means they have not received any modification. X-inactivation center is untouched. Whereas in transgene, you have X-inactivation center inserted onto some autosome. You can see here, this is BRDU, this is a nucleolus staining red color, and then you have got exist. Now it is attached onto chromosome number 8 exist in this experiment, whereas in the bottom panel chromosome number 8 is normal. It is not been done with anything. So wherever you see red color dot, that indicates the nucleolus, and wherever you see this green color dot, that indicates X-inactivation specific transcript is present. So once you merge them, you realize whenever you put exist or X-inactivation center totally in chromosome number 8, it has got a perinucleolar association, but no such association you can see if you have a marker for chromosome number 8. Both the chromosomes are seen properly. Here, remember, here in this control, the chromosome number 8 is not tagged with X-inactivation center, hence it doesn't have X-inactivation specific transcript, but it has some marker for chromosome number 8, that some other gene expressed specifically to chromosome number 8. So what you should understand, chromosome number 8 do not have an affinity for nucleolus, but the moment you put X-inactivation center in chromosome number 8, you will have its affinity going closer to the nucleolus. So what it tells you that during the inactivation process, the X chromosome, which normally have the actual exist or actual XIC, X-inactivation center, will associated with nucleolus is not by randomness. The credit goes to the X-inactivation center itself. So, and this might be contributing for the peripheral localization, eventual peripheral localization of the X chromosome. So let us see other supporting results. Exist is essential for perinucleolar localization of X-inactivation center. Loss of exist, less heterochromatin formation, means you will not have H3K27 trimethylation effectively. So what it indicates that X-inactivation, the guide or the inspiring force of X-inactivation center is exist itself. So dissociation from the nucleolus results in the erosion of repressive chromatin marks. So what researchers have shown that if you try to disassemble that, let this X chromosome, inactive X chromosome, be not allowed to interact with nucleolus, then the maintaining becomes little difficult. It's just like a newborn baby is separated from the mother. Of course it may survive, but it will not survive as effectively as a newborn baby survives with the mother. Same way, if you prevent the nucleolar association, then the inactive X chromosome need not stay inactive. It is vulnerable to get active, vulnerable to get into active phase. So dissociation from the nucleolus results in partial X reactivation. So what it indicates, the inactive X chromosome's association with the nucleolus is not by chance but an obligation. It is necessary as a requirement. It is interacting, and if you prevent that interaction by chemical tools or by genetic tools, you can make the X chromosome, inactive X chromosome, to reactivate partially. So let us see in this cartoon a model for X inactivation. So you have pre-X chromosome inactivation. That means both X chromosomes are given a equal strength, equal bias to express. That is pre-X chromosome inactivation. Once X chromosome inactivation is onset, then there has to be a pairing. Like you can hear, they are separately present. This blue color and yellow color dots are nucleus and nucleolus, and they are now counting. They are paired. Now XX pairing is happening, and their counting happens, and then the choice of silencing also happens. Then what happens? The perinucleolar association in the mid-late S phase takes place, and this has to be occurring, or this has to be preferred for maintaining the X chromosome inactivation. If nucleolar association is disassembled, then maintaining the X chromosome in inactive state can be problematic. So this is the key, that nuclear membrane in black color, and blue color is the nucleus, and the yellow color is the nucleolus, and this green color dots are exist, and also inactive X chromosome, and red color dot is the X inactivation center, and the green color oval shape is XA. So what happens later is decreased perinucleolar association in G0, G1 and early S phase or G2 phase can be seen. So eventually they will have decreased perinucleolar association, because this is required for the division process, for the further replication, because even an inactive X chromosome, it has to be allowed to divide, because it has to unpack by late S phase. It has to be allowed to, it has to be given a choice to divide. So to summarize, the X chromosome silencing via spatial and temporal compartmentalization. It's not just exist alone is required, it also has to merge or co-localize or go to the vicinity of the nucleolus to maintain that. In perinucleolar space, X inactive means XI or inactive X chromosome, and other heterochromatin could replicate. That is a place where they replicate, and exist expression targets autosomes to the perinucleolar space, but they may not be getting silenced as it is occurring with the X chromosome, in at least in that experiment where they tried chromosome number 8. Exist deletion, what happens? Loss of heterochromatin, transcription upregulation is seen if you delete the exist. So how does exist lead to the localization of X inactive or inactive X chromosome to the perinucleolar ring? So this remains like a big question. So this picture we have seen it already. So let us see how X chromosome causes the inactivation in a XI. The exist is expressed from the X inactivation center, and it spreads from the nucleus point, and it brings down, it causes the condensation, brings down these line elements closer, and exist RNA coats the entire chromosome, and it brings a establishment of a inactive state, and it also finally recruits specific macro H2A recruitment, the specific type of histone, and it seals or finalizes the compaction. Now let us see another article which discussed about the role of a RNA, that is role of this exist RNA tethered onto the X inactivation center by a protein called yin-yang, that is called YY1. So YY1 is capable of tethering this inactive exist RNA, or for causing the X chromosome into inactive phase. So exist RNA need to be coated, some glue, someone has to stick this exist onto the X inactivation center. So this is facilitated by a protein called YY1. So how can we prove it? YY1 is required for the X-cyst localization. So we can see X-cyst expression in green color. This is a RNA in situ hybridization, and you also have a situation. This is scrambled siRNA in this whole set. And here you have got YY1 specific siRNA. What is siRNA? We know that it is used for blocking the expression. That's called gene knockdown. So if you use siRNA against any gene, that particular gene production or the particular RNA's translation is prevented. Why? Because siRNA will cause the degradation of that mRNA. So you will not have the protein. So in this experiment, you have scrambled siRNA. That means it is not targeting any mRNA, it is random. Whereas this is targeting the YY1 mRNA. As a result, the YY1 protein is not formed. So in this section. And the bottom panel is the DAPI that marks the nucleus. So blue color one is marking the nucleus. So you can see when you use scrambled siRNA, you are having a S3K27 methylation present normally. Whereas if you have YY1 siRNA, you do not have, you lose this S3K27 methylation. And X-cyst staining, if you are doing in scrambled siRNA, if you have both X-cyst that is present, but if you get rid of the YY1 in the YY1 siRNA one, you do not have the X-cyst RNA present. So what indicates X-cyst RNA to go to the center, you need to have the YY1 RNA. So we will go through this importance of YY1 in localizing the X-cyst onto X-inactivation center more in detail in the coming classes. Thank you.